welcome to online NPTEL course on Introduction to Complex Biological Systems, and for this lecture I am going to discuss about cell junctions and extracellular matrix. So mostly I will be covering these major points: the overview of cell-cell adhesion and the understanding various types of cell-cell junction, followed by an introduction to extracellular matrix, and finally the cell matrix junction. So let me briefly discuss about overview of cell-to-cell adhesion. So cell-to-cell adhesion is a prerequisite for multicellular organism like human, like plants. Now, if you see unicellular organism, like I would say some bacteria or even some unicellular eukaryotes like amoeba. So in those systems, whenever the chromosome or the genome, they are getting replicated, and then if nutrients and all other factors inside the cell they are like fine, then the cell will divide and it will give rise to two cells. If we consider about bacteria, so it will just whenever the chromosome will duplicate, and then it will give rise to two bacteria after division. And this is same for unicellular organism in eukaryotes also. But for eukaryotic system where organisms are multicellular, for example human, it's much more complex. All of us, we started from single cell, say we are starting from zygote, right? So now when it will divide, and finally I mentioned that in adult human being approximately we have 10 to the power 13 cells, but almost all of those cells are somehow attached to each other in some ways. So that's why cell to cell adhesion is very important for multicellular organism. And not only multicellular organism, I would say even for some prokaryotes, for some bacteria, it is sometimes very important. When some bacteria, they infect us, they colonize in human body, they also need to attach to the host cell surface, right? So they get attached to some kind of molecule present on the host surface. So this is also cell to cell adhesion, but this is some kind of pathogen and host interaction. Pathogens are getting attached to the host cell, right? But mostly in this lecture I will be discussing about cell to cell adhesion in context to our own body. Mostly I would say in mammalian system, in human system, how cells are adhered together. That is the major focus of today's class, right? So as a result of that, I would say here I mentioned already that 10 to the power 13 cells approximately. If you consider some cells, for example some immune cells, they are kind of traveling through our blood and other fluids. So as a result of that, they are not constantly adhered to some particular sites, but they also dynamically they adhere as and when required. So as a result of that, you can understand that cell to cell adhesion is also very dynamic process. So here we are going to discuss about mostly cell to cell adhesion and cell to matrix adhesion in context to mostly animal cells, mostly mammalian cells I would say more specifically. Let's see. So the adhesion properties of epithelial and connective tissues. So epithelial cell means for example during our last discussion, during the last lecture, so during that time we discussed about intestinal epithelial cells, for example. So if you see any epithelial cell, so here, so these are epithelial cells. As you can see, this is the epithelial cell layer. And now those epithelial cells, they are attached to each other. So you can see here, this is the attachment site. I would say if this is the cell 1 and this is cell 2, you can see they are attached here, and some junction, some cell to cell junctions are present here also. And in addition to that, these are this red fiber kind of thing, these are nothing but cytoskeleton. So you can see here the mechanical stresses are transmitted from cell to cell by cytoskeletal filaments. So cytoskeletal filaments means here it can be actin filament, it can be intermediate filament, which you already learned in different lecture in cell. Now, so as you can see, in our body, different types of tension, different type of stretch and pulling force, and different types of force sometimes generated because of our movement and so many other reasons. But during that time, this mechanical stress can be tolerated. So like what I am trying to say, in this case, in case of epithelial tissues, these cytoskeletal filaments which are present and they are anchored to cell matrix and cell to cell adhesion sites, they are the major load bearing thing. On the contrary, if you see in case of connective tissue just beneath the epithelial cell layer, so as you can see, this is basal lamina on which our epithelial cells are attached, and just below it we have some connective tissues, right? So in connective tissues you can see cells are staying in a little bit different way. Not those cells are very much compact and they are not very much attached to each other. As you can see here, so this is one cell, right? This is one cell, another cell here you can see, and those cells are actually surrounded by some extracellular matrix, which I will discuss in more detail. So now in this case, in case of connective tissue, the extracellular matrix directly bears mechanical stresses of tension and compression. Okay, so this is the major difference between epithelial tissue and connective tissue. So example of connective tissue, I would say some kind of, tendon, bone, those are example of connective tissue. And epithelial tissue, as you already know, like one example is intestinal epithelium and our skin layer, and many other portions of our body, they have epithelial tissues. Now a little bit more detail about cell to cell and cell to matrix junction. So first of all, I should little bit explain what is matrix, because when I was showing that epithelial layer of cells, those cells are just attached one by one, right? But below that layer we have some kind of matrix, and those matrices are nothing but mostly defined types of protein and carbohydrates which are secreted by cells, particularly cells present in connective tissues, and they form some kind of gel-like structure. Sometimes fibrillous proteins are also there. All together they form extracellular matrix or cell matrix. Now if you see here, here I would say this is cell number 1, cell number 2 and cell number 3, as you can see, and this is again some epithelial cells and they are present consecutively. Now if you see those cells, for example cell 1 and cell 2, so this side is apical side and the basal side which is getting attached to the basal lamina. So this is our basal lamina and this is the apical side. and this is basal side. Now, from the top, if we see, from the apical side, if we see, like at the very beginning, we have this kind of junction which seal those cells together. So that, for example, if I consider this is intestinal epithelial cells, then this part should be the lumen of the intestine. So we have some fluids, some nutrients, and so many things. So then, if those cells are not sealed through tight junction, then some material can enter through this like space between two cells. So that's why tight junction, they basically seal this gap. So this is tight junction. Followed by, we have two different types of junction. Those are adherence junction, this one, and desmosome, this one. So together, like adherence junction and desmosome, together we say cell to cell anchoring junction. They somehow anchor two cells together, right? So if you see adherence junction, like some proteins are involved there, I will discuss in more detail soon. So those proteins from those two cells, from the adjacent cells, they actually interact through their extracellular domain, and through their intracellular domain, they interact with the cytoskeleton. So as you can see, in case of adherence junction, whatever proteins involved there, they interact through actin filament. Similarly, in case of desmosome, if you see, whatever proteins involved there, they interact through their cytoplasmic tail to intermediate filaments. So as a result of that, those two cells are now physically attached to each other through this procedure. And particularly, as I already mentioned, this adherence junction and desmosome, these types of junction together, we say anchor junctions or anchoring junction. And in case of adherence junction, the major proteins involved there are classical cadherins. So cadherins are a very big group of cell adhesion molecules. We have in our body almost more than 100 different types of cadherins, and they are majorly categorized into classical cadherins and non-classical cadherins. And in case of desmosome, cadherins are non-classical cadherins are involved in this desmosome. Now we have another type of cell to cell junction, that is called gap junction. As you can see, this is gap junction. So here they are basically channel forming junction. So the previous two, adherence junction and desmosome, their main property is to keep those two cells together physically, and like through those protein, they are finally attached to the cytoskeletal material, as I already mentioned. But in case of channel forming junction, the example is gap junction. From the name you can understand some gap is there. So those two cells are directly communicating with each other. They can send some small, like water soluble molecule from this cell number 2 to cell number 1, for example. Similarly, from cell number 3 to cell number 2, they can directly transfer some of the small molecules, not very big thing, not very high molecular weight proteins or nucleic acids, because of the pore size. I will discuss in more details later, but this is gap junction. And apart from this cell to cell junction, whatever I mentioned, 4 types of cell to cell junction I already mentioned. One is tight junction, adherence junction, desmosome, and gap junction. And also we have some cell matrix junction. So as I already mentioned, that the epithelial layer should be attached to some substratum, some kind of layer called basal lamina. So as you can see in this case, so for example, this cell matrix junction, this is actin linked cell matrix junction. So some proteins are again linked to the actin cytoskeleton present inside the cell. So some protein present in the extracellular matrix, right, that is interacting with some other protein present on the cell surface, and finally it is getting attached to the actin cytoskeleton. Similarly, in case of hemidesmosome, this is again like intermediate filaments are involved in this case. So that way, I hope that you understand that cell adhesion means cell to cell adhesion and at the same time cell to matrix adhesion. Now, very briefly, I will discuss the mechanistic basis of anchoring junction. That means particularly I will focus on adherence junction that support cell to cell adhesion. So all these 4 types of cell junction I already explained. So if you see here something like this, I would say this is one cell and this is another cell. So now what happened? Cadherins. So cadherins, like particularly classical cadherin, they look like this. They have 5 extracellular domain, as you can see here, 1, 2, 3, 4, 5. So 5 extracellular domains present in cadherin. So this is classical cadherin. And another cadherin here, for example, if I say this is cadherin, like something cadherin 1, cadherin 2, some cadherin molecule coming from the next cell, but cadherin should be kind of same cadherin, like same kind of cadherin molecule should be involved, right, in this interaction. So here classical cadherin. So when they are interacting through their extracellular domain at the cytoplasmic site through some adapter proteins. For example, these are the adapter proteins. Adapter proteins. They will be finally attached to the actin cytoskeleton. Through adapter protein, they will be finally attached to actin. Here, actin. So now I hope that you understand that how these interaction through cadherins to their extracellular domain finally help to adhere these 2 cells together. But only thing in the cell junction site, not just one cadherin from this cell and the one cadherin from the adjacent cell they interact. Many cadherin molecules together they assemble, they form a very large complex, and thereby they can support cell to cell adhesion. Here, cadherins mediate homophilic adhesion. Homophilic means here I would say like same cadherin interact with like I told that we have more than 100 different types of cadherin. Like for example, I would say like major cadherin, classical cadherin, for example, E cadherin. This is very common, E cadherin. So here E cadherin means this is epithelial. E stands for epithelial. Okay, epithelial cadherin. Because it was first discovered in epithelial tissues and it is also predominantly present in epithelial tissue, E cadherin. Similarly, we have N cadherin, which is mostly found in neuronal tissue, mostly on nerve cell. It is also present in some other tissue, for example, in heart muscle also we have N cadherin, but it was initially discovered in neuronal cell. That's why its name is N cadherin. Similarly, we have P cadherin, which is present in placenta, and R cadherin, which is predominantly present in our retina. So these cadherin, they interact in a homophilic fashion. That means E cadherin interact with E cadherin. Similarly, N cadherin interact with N cadherin, and that way they support cell to cell adhesion. Here you can see that this is adherence junction, already I explained, and these 2 cadherin, although you can see here in different color for the clarity, I would say this is E cadherin and this is also same molecule E cadherin, but they are coming from 2 adjacent cells and they interact through their N-terminal extracellular domain, and on the C-terminus they should be attached with some actin cytoskeleton, okay, actin bundle. Similarly, here also they will be attached to the actin cytoskeleton. So that way cadherin mediate this homophilic adhesion, okay. And I already mentioned that cadherins, they are the major group of cell surface protein, and particularly they are glycoprotein, means some carbohydrates are also attached to this protein. That's why they are called glycoproteins. So during translation we get the polymer of amino acids, but after some post-translational modification, some carbohydrate groups are also attached to this protein for their some special function and also for their stability. And another important aspect about cadherin is that their function is dependent on calcium. So calcium is very important for their function. As you can see, this is how cadherin looks like. I would say this is, again, if I say this is E-cadherin. So E-cadherin, N-cadherin, they look very similar like this. They have 5 extracellular domain, this 1, 2, 3, 4 and 5, and in between those domains they have calcium. These red dots you can see, those are the calcium ion, and it is very important. If you deplete calcium, if you take out calcium, then the cadherin structure will collapse and they cannot mediate cell to cell adhesion. And even this name came from this cadherin, the first 2 letters here, this is because of the calcium, like they depend on calcium. And during the discovery, when scientists understood that they only work in presence of calcium, and that way it has been discovered, they got this name cadherin. Now, some interesting experiment I am going to discuss about cadherin and how it helps in the development of tissues. Like cadherin dependent cell to cell adhesion guides the organization of developing tissue. In this figure, whatever you can see now, this is sorting out of cells. So what scientists did, they have taken cells from early amphibian embryo. Amphibian embryo means some, I would say, frog embryo or salamander embryo, but early embryo. They have taken those cells and somehow they mix those cells together, okay. So after taking those cells, they just mix it together. So as you can see here, this is the initial point of the experiment. Cells are mixed together here. Cells are mixed together. And here, as you can see, that these red cells, they are epithelial cells. And these green cells, as you can see here, these are mesoderm cells, and these blue cells you can see here, for example, this one, many blue cells are present here, right? Those are, I would say, neural plate cells. That means part of our, part of developing neuronal tube. You can consider that way. Neural plate cells, they are mixed together. And now, when those cells are there and like they were allowing some time, you can see somehow the organization is slowly changing. As you can see, those cells are getting sorted out by themselves, right? And here you can see this is almost the reminiscent of the embryo. Almost it looks like embryo structure. Why? Because as you can see, after all these steps, you can see all those epithelial cells now, those are epithelial cells, they are on the periphery. Similarly, the mesoderm cells present just beneath the epithelial cell layer, and neuronal tube cells, the neuronal plate cells, they are inside. This is neuronal cells. So as a result of that, which is like true for during the development of frog embryo itself, then how it happens? So, you know, major, like I would say, like a lot of research actually like found that this is because of cadherin itself. Cadherin help this sorting out of those cells, and they organize the developing tissue in this way. So, like if I explain this figure, you will understand even more, you know, clearly. As you can see, this is the ectoderm layer, and then you can see during the neural tube formation. So here we are trying to explain the cadherin expression pattern, how it guides to the construction of the vertebrate nervous system. So as you can see here, that the neural tube formation is taking place. During that time, the similar type of cadherins, they are staying together, and finally the ectoderm layer is getting separated, and you can see this neuronal tubes is already formed here, right? And here this is kind of in color code, we are mentioning different types of cadherin like, you know, majorly present in these cells. As you can see, the ectoderm cells, they have very high level of E-cadherin, and you can see that E-cadherin, they are getting like, you know, present in these ectoderm cells, and this is getting separated from the neuronal tube. And in the neuronal tube, mostly you have what? N-cadherin. I already mentioned that N-cadherin is majorly present in neuronal tissue. So as you can see, this is yellow color code here. So this is almost everywhere, this is yellow, right? So neuronal tissue. So this is the brief idea how like, you know, cadherin helps in tissue organization. And the basic idea, I would say, like from where this is coming, this is very simple experiment someone can perform, that is something like this, that cadherin dependent cell sorting. If I say that those cells which I am drawing now, this black color cells, those are E-cadherin expressing cells, E-cadherin expressing cells. And now these cells, which I am not making it, you know, I am not filling it. So this is N-cadherin expressing cells. Now if you mix together these two cells, and then you will be seeing, like, you know, you mix together, okay? So as a result of that, you can say something like this scenario. It's everything mixed together, okay? Everything mixed together. N-cadherin expressing cell and E-cadherin expressing cell, but it will be sorted out soon. Those E-cadherin expressing cells, they will form a cluster. They are expressing E-cadherin, and N-cadherin expressing cells, they will be sorted out also. This is the basic logic of those things, whatever I have just explained in these two different types of scenario, okay? Now, what is happening inside the cytoplasmic side, like when cadherin they interact? So as you can see here, another group of proteins in the, like previously I mentioned some adapter proteins, because, you know, sometimes it's very difficult to remember so many names there. That's why I just mentioned adapter proteins. But those are the catenins. Catenins link classical cadherins to the actin cytoskeleton, right? So this is cell 1 and cell 2. And now if you say this is one cadherin, this is another cadherin, now they actually interact with this actin cytoskeleton here. As you can see, this is actin, okay? This is actin, and through the cadherin directly cannot bind to actin cytoskeleton. They need some adapter proteins, and those adapter proteins here, so these are adapter proteins. adapter proteins. So those are, for example, different types of catenin like P120 catenin, beta catenin, alpha catenin. Nothing will happen if you forget those names. But overall idea, that's why I'm drawing here, that when catenins, they are interacting through their extracellular domain, at the same time through their cytoplasmic domain, through some adapter proteins, they are physically attached to the actin cytoskeleton. Thereby, two cells are interacting together. And catenin-catenin interaction here, catenin-catenin interaction, generally it's very weak interaction, but it's dynamic interaction. So that's why to support this weak interaction and to get it, physiologically relevant so that cell can be attached together, cell can be adhered together, many catenin molecules, they assemble together and thereby they perform physiological function. That's all. Now, like whatever I mentioned, that is about mostly anchoring junction. I explain more about adherent junction, but this is almost like same thing for desmosome also. That is also one type of anchoring junction. But now I am going to discuss about the tight junction. So it is also very important, not just keeping cell together, they are attaching those two cells together at some places, but particularly on the apical side of the cells. And in addition to that, they also play important role in cellular transport. That is very important. Again, this example you can consider in context to intestinal epithelium. So I am taking intestinal epithelial layer as model organ, model tissue system to explain this thing. So it will be easy for you also, like if I say kind of similar thing and different types of explanation, different types of scientific observation I am going to discuss with intestinal epithelial cells. So now, as a result of that, you can see this is lumen of gut. So now you can see through apical side, here you have some molecules here, nutrients, water, all those things present inside the intestine. But they can easily pass here. In between two cells, we have some space in between two cells. But this material present in the lumen, they should not enter in between cells. So that's why I would say tight junction is present. Tight junction somehow seal this portion. You can see here, they are sealing this portion, So nothing can enter directly in between two cells. So then that way they are helping. And now I just mentioned that tight junction, their role in cellular transport. So as you can see, like through intestinal epithelial cell, we absorb most of the thing, almost everything through intestine, from intestine. So here particularly we are talking about glucose transport. As you can see that glucose is also present in the lumen of gut. And now we have some sodium driven glucose transporter on the apical side. So this is our apical side and this is our basal side. Okay. So you can see some sodium driven glucose transporter. So as a result of that, glucose will enter here, glucose will enter here. So as you can see, glucose there, and glucose concentration will be high, right? Now this glucose, like those are the absorptive cells, they are absorbing glucose, they are taking glucose from the intestine. Now this glucose should go into the other part of our body. So as a result of that, you can see here the glucose concentration is maximum in this epithelial cell, high glucose concentration here. And now this high glucose concentration, this glucose will slowly diffuse towards other side. But here you have sodium driven glucose transporter. So as a result of that, this glucose will not again get out into the lumen of the gut lumen. No, that is not possible. But it will diffuse to the lower side, I would say toward the basal side, and finally it will go into the like in a bloodstream, and glucose will be carried over. Similarly, some passive glucose transporters are also present in this cell membrane, the basolateral cell membrane. So this is basolateral, the basal and the lateral, that's why basolateral cell membrane. So as a result of that, some glucose molecule will come here in this passive glucose transporter, and through there it can also enter, it is possible to enter here. So now directly from lumen, gut lumen to these cells, and from these cells is passing and then it going to this blood. So that way it is crossing these cells. This is, we say, transcellular transport. And in the same layer, when this way it's moving, right, this is paracellular transport, transcellular and paracellular transport. That is just some kind of information that is not that much important to understand cell adhesion itself. Now, what is the mechanism in case of tight junction formation? So as you can see, instead of cadherins, here you have different types of proteins. This is called like tight junction protein. Particularly these two proteins are very important, claudin and occludin. One very fundamental difference between this cadherin and these proteins. Cadherins, they cross the membrane just one time. They are single pass transmembrane protein. But here you can see like they are passing 4 times, like 1, 2, 3, 4. So they are passing this cell membrane 4 times. So they have multiple membrane spanning domain, like spanning region. So this is our cell membrane, and similarly this is also another cell membrane. This is cell 2 and cell 1, and this way tight junction is formed. Now the next thing is gap junction. So gap junction couples cells both electrically and metabolically. Why? Because gap junction means here those two cells are directly connected through each other by some kind of channel. As you can see, this is direct channel. Okay. So something can go from this cell to other cell. Particularly I mention here electrically and metabolically because here lot of ion, for example Na plus, chlorine minus, and so many ions can move, and that's why we are mentioning that electrically. And metabolically, small water soluble nutrients, for example some glucose molecule, like small, I would say amino acid, nucleotide, they can cross through this channel or through this gap junction, right? So like these are the ion, and then as I already mentioned, amino acid, sugar, they can quickly go, no problem. But if something is very big, I would say above 5,000 Dalton, 20,000 Dalton, they are much bigger to this pore size. So as a result of that, they cannot cross. So as a result of that, a big protein, for example protein or nucleic acid, they cannot move through this pore. Only small nutrients and ion can move. And as you can see here, this is just in a different dimension. This figure you can see, this is one plasma membrane and this is another plasma membrane, and this is the channel here. As you can see, thing can move in this direction, right? So those are the channel, and these are the channel forming protein. The protein which form these channels are called connexins, particularly in case of vertebrates, in case of mammalian system. So these connexins, they form some kind of I would say hexamer. As you can see, 1, 2, 3, 4, 5, 6 connexin, they are together and they form some kind of hexamer. So it can be homomeric, that means same kind of connexin they are involved, or heteromeric, that means different types of connexin involved together, and those two connexin, they are forming this channel. This is the channel. That means this channel, for example this one, if you just rotate 90 degree, you will get this. So this is from one cell and this is from another cell. So cell 1 and this is cell 2. That way they form this kind of channel or some kind of pore, so that things can go in and out between these two cells. and this is the extracrystallographic structure of this channel. As you can see, this is the pore dimension. The you can see here is 1.4 nanometer the diameter here. So as a result of that, restrict the movement of bigger stuff in between those two cells. And now I'm going to discuss about little bit different thing here. Whatever cell-to-cell junction I mentioned, like I would say tight junction, then adherence junction, desmosomes, and gap junction, those are kind of more stable cell-cell junction, right? But some other cell adhesion also happen, which is more dynamic in nature. For example, selectins, this is another group of cell adhesion molecules. They mediate transient cell-to-cell adhesion in the bloodstream. Like those cell adhesions are very transient in nature. So selectins, like in this case, cadherin, they interact with another cadherin, they interact with another protein, right? But selectin, you can see, selectin are the cell surface carbohydrate binding protein. So selectin, they specifically interact with carbohydrates. So that is their kind of specific feature, right? So for example, there are different types of selectin also present. For example, L-selectin is present in white blood cell. Similarly, P-selectin is present in platelet, E-selectin on endothelial cells, something like that. And now, overall idea I would like to present, not this just name that E-selectin and P-selectin. So the idea is here, the adhesion should be transient. Why? So for example, you know that our immune cells, particularly if we say WBC, white blood cell, they are kind of, I would say, they perform some kind of bohemian life. So they are not restricted to a particular region. So they are like, traveling throughout our body. Whenever some requirement happens, some infection, some particular site, they can go there also. So as a result of that, they are more dynamic in nature and they rely on selectin, this kind of cell adhesion molecule. So as you can see, this is the lumen of this blood vessel, and this is not epithelial cell anymore. This is endothelial cell, which is covering the, I would say, the blood vessel from the inner side. This is the endothelial cell. Now these white blood cells, they have selectin. Selectins are present in this endothelial cells. So I would say this is selectin. Selectins are present on endothelial cells. And white blood cells, they have some specific type of carbohydrates, right? They are present on WBC. And that carbohydrate and this selectin, they interact together. But this is not very tight adhesion. This is very weak adhesion. Still, those attached white blood cell on the endothelial surface, they can roll. They can go in some kind of rolling motion. And at some point of time, when integrins present on white blood cells, they bind to the another group of cell adhesion molecule, which are little bit different family member. I'm not going into their details, name and all those things. And that will help to that cell, particular white blood cell, to come out, or I would say to come into the tissue. As I already mentioned, that white blood cell, they also escape this bloodstream and they go to the tissue for their own function, for their immune function. So as a result of that, during that this in and out, for their function, they rely on different types of dynamic adhesion. So that is all about, I would say, selectin and their transient cell-to-cell adhesion properties. This is just one example. There are many other examples. Now, at the beginning, I mentioned that I will little bit talk about the extracellular matrix also. So this is particularly extracellular matrix of animal cells. Like as you can see, this is fibroblast in connective tissues. So this like fibril-like structure, as you can see, like thread-like structure, these are actually, I would say, all these things are collagen fibrils. So collagen, they are actually protein, some kind of protein fiber, and they are also giving us a lot of support in our, particularly in our connective tissues. they are present in the extracellular matrix. Those are actually generated by cell itself. Cell secrete this protein, and this is collagen fiber. And apart from collagen fiber, we have some carbohydrates also, for example, hyaluron, right? So, and hyaluronic acid, and then proteoglycan, all those things together, they form this extracellular matrix. So, as you can see here, this is hyaluron. So, this is some kind of carbohydrate, as you can see, the repeating disaccharide units, and this is also present in large amount in this extracellular matrix. And also another very important thing is collagen fiber, which I already mentioned. So, in case of mammalian system, in our body, I would say collagen, this is a protein fiber, but collagen is almost, I would say, 20 to 25% of total protein present in our body. And especially collagen, they have some kind of typical structure. They are kind of triple helix. So, 3 collagen strands, they are forming some kind of helix, as you can see here, triple helix of collagen. 3 collagen strands, they are kind of forming some kind of helix. And collagen, they have some other properties also. This is the collagen strand, you can see here. This is collagen strand. Sometimes it is called alpha chain of collagen, but here you can see like some kind of repetition here, that X, Y and glycine. So, X is really its proline, Y is hydroxyproline, and then this is glycine. So, then it is repeating in this way, and that is kind of major, I would say, it's give us lot of mechanical strength and support in our tissues, right. So, this is about our extracellular matrix, and I believe this is the last slide, that now the last thing is the integrins are transmembrane protein that link the extracellular matrix to the cytoskeleton. At the very beginning I mentioned, so in context to epithelial cells, if you consider, those cells are attached together by different types of anchoring junctions, but those cells should be also attached to the basal lamina, otherwise that layer will peel off from the surface, from the connective tissue. So, they should be attached to the basal lamina. So, as a result of that, everything should be together. So, that is why as a result of that, like some kind of cell to matrix junction should be also there. So, here you can see, for example, if I mention here, this is in case of connective tissue, not even epithelial cell. So, this is connective tissue and this is one cell. So, this is the nucleus, another cell, and this is the nucleus. So, in connective tissue, those 2 cells might not be together, but they are somehow attached to the extracellular matrix. So, in the extracellular matrix, you have, for example, collagen fiber, right? Collagen fiber present, and also we have like some other like proteoglycan, hyaluron, and everything present. But here, what I am trying to say, that in these cells, on this cell surface, you have some protein which is called integrin, and this integrin is attached to the protein present in the extracellular matrix. So, that way this cell is attached, or this cell is staying together with the extracellular matrix. This is very important, just like cell to cell adhesion, it is also very important. So, that is why I provide you this context, so that you can understand this figure itself. So, you can see this is one cell, this is one cell, and this is the cytosol, and this is cell membrane, and now you have this integrin. So, like beta subunit of integrin and alpha subunit of integrin, like I should mention here that integrin, they form some kind of heterodimer. 2 different integrin, they form heterodimer, and then they interact, they bind to extracellular matrix protein, for example, some collagen fiber, like this is the extracellular matrix protein. So, as a result of that, connective tissues, for example, they are attached or they are staying together with the extracellular matrix. So, This is, I would say, like all about kind of cell to cell and cell to matrix addition. You have like some kind of idea and now, and for more details, you can follow this textbook, Molecular Biology of Cell by Albert. Thank you very much.